Yeah, good morning, all. Hope you had enjoyed the previous day sessions. Today we have 3 sessions from Dr. S. Balaji. The first session will run through Dr. S. Balaji. The second one will be from Dr. K. M. Kumar. And we have the last session from Dr. Harshavardhan Tiwari. Dr. Balaji will be delivering about the bioinformatics and computational tools for next generation sequencing data analysis. And Dr. K. M. Kumar will be highlighting about the computational tools in tracking COVID-19 outbreak. And Dr. Harshavardhan Tiwari will be giving the explanatory data analysis on PIMA data set, early prediction of type 2 diabetes mellitus. So most of them are hands-on here. So make best use of it. I hope all of you are ready with all the prerequisites, what was put up in the WhatsApp group, also in the blog, you might have noticed, and you're ready with it. I welcome you all for today's sessions. Have a fruitful day. Welcome you all again. My colleague Harshita will introduce Dr. S. Balaji. Harshita.
Good morning, all. I welcome you all to the 2 sessions of this 5-day virtual FDP on potential research avenues on computer science and biology. Today, I am going to give a brief introduction about our resource person, Dr. S. Balaji, sir. Dr. S. Balaji, being an entrepreneur, he himself is the founding director of Chain Technologies Private Limited, whose focus is on developing enterprise solutions using the cutting edge technologies like machine learning, IoT, blockchain, and Agrigenium Innovations Private Limited, which specializes in converting agriculture residues into wealth. Dr. S. Balaji sir is a technologist turned academician with a blend of academic and industry experience with an expertise in successfully executing and managing R&D intensive projects. Sir holds a PhD from Faculty of Engineering specializing in the area of fault-tolerant and real-time systems in computer science and engineering from Indian Institute of Science. As a technologist, Dr. Balaji sir has several accomplishments. He has defined and established software processes for developing mission and safety critical space applications. He has introduced numerous innovations for the design validation of spacecraft, onboard subsystems, and developed advanced test systems to validate the satellite control systems. And also he has mentored the staffs to deliver high integrity systems that meet critical spacecraft mission requirements. Dr. Balaji sir took up his career in education after extensive R&D experience in ISRO and joined as a professor at AMC Engineering College. Sir has joined the Department of Computer Science and Engineering at City Engineering College as a professor and worked as vice principal and then as principal of City Engineering College. He has then joined the Center for Emerging Technologies, Jain University, where he has initiated research in the area of bioinformatics, genomics, and molecular modeling and simulation. He is currently with the Center for Incubation, Innovation Research and Consulting, Jyoti Institute of Technology. His current research includes real-time and embedded system, mobile computing, image processing, heterogeneous parallel processing, and bioinformatics. Sir has published over 50 papers in national and international journals and presented over 30 papers in national and international conferences. Uh, Dr. Balaji sir has received grants from various uh, sessions, that is grants by Vision Group of Science and Technology by Government of Karnataka for setting up a genome computing cluster, and also Babel Chain Technologies Private Limited has secured rupees 50 lakh grant from Biotechnology Industry Research Assistance Council for the development of agri-tech product. Today, Sir will be delivering lecture on the topic Bioinformatics and Computational Tools for Next Generation Sequencing Data Analysis. I welcome you, sir, for the session. Over to you, sir.
The bio and life sciences, and therefore because I'm told that most of you are from computer science background, we may find it a bit difficult to get in terms with the terminology and processes. Nevertheless, it is not that difficult to catch up. We will be able to do. That's why when you go through the slides, and the pointers are provided almost everywhere in every slide, where you can get more details about the various processes and the terminologies that are used in the NGS data analysis. And the theme of this FDP is, what are the kind of research avenues, for computer science and biology. When you take into that, you can see almost in every slide that, we'll see some challenge. Though I won't say that, it is a research topic or a research area, but every challenge is an opportunity for us to get into research and do. That's how, we'll have to look into. And you can see that there are plenty of challenges, both in terms of handling the data and archiving the data, and then developing various algorithms to do the processing of this NGS data. Yeah, next please. I'm sorry, whoever is handling these slides, next slides, we'll have the bullets gone in sequence. Anyway, let me see how we can handle. The agenda is to initially look into what is next generation sequencing, and we have basically now we are in third generation sequencing, and we'll look into what we mean by the second generation sequencing platforms and then third generation platforms. Then we'll look into briefly about NGS bioinformatics, where we'll be looking into several tools for primary. Basically, the NGS bioinformatics is divided into three analysis stages: primary, secondary, and tertiary. And we'll look briefly into what are the various tools that are available for us to do this. And then it's not that NGS is panacea for all the issues that we have. It also has certain pitfalls, and we'll look into the pitfalls at the end before we conclude. Next, please. Yeah. Maybe you can. We all know that genetics is extremely important to medical practice, because genetics provides a definitive diagnosis for many clinically heterogeneous diseases, and genetics enables. Oh, I think you have already moved too fast. Yeah, okay. And genetics enables accurate disease prognosis. The prognosis is more important than diagnosis, and that's where genetics help us a lot. Genetics also provides a guidance toward the selection of the best possible options for giving care to the patient. That's what kind of treatment plans you can have. All those things can be guided using genetics. The current potential of genetics is derived from the capacity to integrate the human genome at different levels. That is, we can interrogate the human genome at levels starting from the single base alteration, that is the changes that take place in single bases, to the chromosome level. Up to that level, it's possible for us to interrogate using the available tools, and therefore the power of NGS data analysis is being utilized in many of these things, especially the Mendelian diseases, which are inherited diseases. The pioneering works on DNA sequencing made possible several progresses, resulting in the so-called Sanger sequencing, which we will not be looking into. That is basically the first generation sequencing, which was followed by several automated DNA sequences that facilitated the sequencing of the complete human genome in 2001. It was in 2001 that the human genome was completely sequenced, and that's when a lot of opportunities have started pouring in. Next, please. I think somebody in between. Can you please mute? Can you please mute your audio, please? Somebody in between is talking.
Sorry, sir. Unmute yourself, sir.
Can I get back this slide, please? Yeah, right. Thank you. Now, if you look at the timeline of DNA sequencing, see, though the concept of DNA, I think got disturbed. Yeah, okay. The concept of DNA is not new. Even it was there in the 19th century. I think sometime in 1894, the DNA has been kind of the concept has arrived, and it is in 1953 the discovery of DNA structure was done. I think the screen is jumping. Yeah, please can I hold it like that? And in 76, first genome was sequenced. It was for bacteria, and the publication of Sanger sequencing method was done by Sanger, who got Nobel Prize twice. And then the PCR development, polymerase chain reaction technology was developed in 83. 86 saw the launch of the first automated sequencer. These are all the tools which have helped in sequencing the human genome, and complete sequencing of E. elegans, which had 100 million base pairs, happened in 98. And by 2001, we were all equipped. The scientists were equipped to sequence the complete human genome, which has about 3.2 billion base pairs. We'll look into what a base pair. Anyway, I think you would have seen yesterday what a base pair is. And then the NGS, that is NGS stands for Next Generation Sequencing. The Roche 454 NGS system was launched in 2005, and then 2006 another sequencing machine was launched. 2009 a single molecule sequencer was launched called Helicos genetic analysis system, and then PacBio has launched in 2001 another sequencing machine. And if you see, up to 2006, they were basically second generation sequencers, and then started coming the third generation sequencers like PacBio. PacBio is one thing, and Oxford Nanopore is another thing. Then SeqLL is another sequencer that is in third generation. Next, please. So we are in third generation now, and the latest third generation sequencer was launched in 2017. Several new approaches were targeted to complement and eventually replace the Sanger sequencing. The reason was Sanger sequencing was taking too much of time, and this technology is collectively, because all these technologies that were introduced to replace Sanger sequencing technology combined, is known as next generation sequencing or massively parallel sequencing. This is an umbrella term to designate a wide diversity of approaches. Through this technology, it is possible to generate massive amounts of data per instrument run in a faster and cost-effective way. That is the real need as such, because as we have seen, the human genome has 3.2 billion pairs, and accordingly, if you see the amount of data that you generate when you sequence the human gene, the human genome, it turns out to be something like of the order of 30 GB of data. Now it is possible to stream in parallel huge data of several genes or even the entire gene in one run. The technology has improved such a way that it is possible to do all these things. Next, please. Now let's quickly, though this is not a sales talk, just to get a feel of the importance of NGS. It's better to get a feel of the NGS market. I think the slide got disturbed. Okay. The global market for NGS data analysis is projected to reach about $21 billion by 2025. This is an annual growth rate of about 20% from 2017 to 25. And there are several brands, because the market is huge. 20 billion plus is not a small market. Since the market is so huge, there are several brands which came into the market. There are these are the top brands in the market: Illumina, Ion Torrent, that is this from Thermo Fisher Scientific, then BGI Genomics, PacBio, and Oxford Nanopore. These are the 5 major brands that are in the market today. All these brands provide different strategies towards the same problem. The problem is the massification of the sequencing data. How quickly you can do the massive data that you get in NGS sequencing, how quickly you can do, how quickly you can organize, and how quickly you can archive, and how quickly you can do the analysis on these data. This is the focus of all these brands. The second generation sequencing is based on massive parallel and clonal amplification of molecules, which is called polymerase chain reaction. These are all the terminologies that we need to pick up at some point when we want to be bioinformaticians. The third generation sequencing relies on single molecule sequencing without prior clonal amplification. That is the main difference as such. There are few advantages of sequencing with third generation, which we will be seeing in the next slide. Next, please. See, whenever we want to do a sequencing, we need to create what is called a library. See, though this is not directly related to the analysis, but we need to know that even in these areas, there is a lot of software tools that are required, and we have several challenges in handling even the NGS library preparation. So you need to prepare a library. A library is a collection of, actually the slide keeps getting disturbed. Is there anything that we can do about it? Yeah. A library is a collection of DNA or RNA fragments that represents either the entire genome or transcriptome or a target region. It's a collection of DNA fragments that we will be using. Each NGS platform has its specificities, but in simple terms, the preparation of an NGS library starts with the fragmentation of the starting material, then connecting sequence adapters to fragments to allow the enrichment of those fragments. So whatever DNA or RNA you have, you are going to fragment it, and then you will add at the ends, to enrich these fragments, and then use them for sequencing using the processes that are followed in NGS sequencing. A good library should have great sensitivity and specificity. All fragments of interest should be equally represented in the library and should not contain random errors. This is one of the requirements of NGS library preparation. And then whenever we sequence a genome, it's not that genome regions are completely prone to be sequenced. It's not possible to make sure that the complete region is sequenced, and therefore the construction of sensitive and specificity is very, very important in preparing the libraries. Yeah, next please. Okay, basically there are 4 steps in preparing the libraries. The first step is to prepare libraries in most NGS workflows is the fragmentation of the DNA or RNA or the so-called nucleic acid. The fragmentation can be done either by physical or enzymatic method. These are the 2 methods that are followed in fragmenting the DNA. The physical methods include acoustic shearing, sonication, and hydrodynamic shearing. These are all the processes used in the NGS processing. Enzymatic methods include digestion by DNase or fragmentase. This is also another process. One of these 2 processes is used to fragment the DNA into smaller fragments. Both methods give similar yields. Anyway, there is absolutely no problem in using either of these methods. The choice of the method that you use between the physical and enzymatic method only depends on experimental design that you do or the external factors such as whether you have the lab facilities to carry out these kind of things. That's all. Otherwise, you can choose either of them. Next, please. The second step, once the starting DNA has been fragmented, adapters are connected to these fragments. Basically, these adapters are introduced to create known begins and ends to random sequences, allowing the sequencing process. Because in the sequencing process, we should be able to identify which is the beginning and the end of the fragment, and therefore you add known sequences to the fragments as adapters. An alternative strategy combines fragmentation and adaptation ligation in a single step, which is called tagmentation. This is another way of doing it. Next, please. The third step is, the third step of the library preparation. We are in third step of the library preparation, and all these stages require a lot of software tools that we need to support with. Upon nucleic acid fragmentation, the fragments are selected according to the desired library size. The size of the library is limited either by the type of the NGS instrument that you have, or the short read sequences such as Illumina and Torrent present best results with the DNA libraries that contain shorter fragments of smaller sizes. If you have shorter reads, it's always better, you get better results. Illumina fragments are longer than that in the Ion Torrent. In the Illumina machine, because we are talking primarily of 2 major brands, Illumina and Ion Torrent, and these are two major second generation NGS machines available from both the brands. In Illumina, the fragments can be up to 1,500 bases in length. In Ion Torrent, the fragments can go up to 400 bases in length. Long read sequences like PacBio, that is the third generation NGS machine, produce ultra long reads by fully sequencing a DNA fragment. So this can, instead of sequencing the DNA in terms of several reads, the third generation machines like PacBio can sequence the entire DNA fragment into a single sequence, single read. The optimal library size is also limited by the sequencing application. Whatever is the application that you use, based on that you have to decide what is the kind of optimal sequencing library size that you want to have. For whole genome sequencing, the longer fragments are preferable, while the RNA sequencing and exome sequencing, smaller fragments are feasible since most of the human exons are under 200 base pairs. The exons are the ones which are from the DNA, they produce the proteins. Next, please. In the fourth step is the enrichment step. The amount of target material is increased in a library to be sequenced. That's what happens in the enrichment. which basically it does amplification and generates from one DNA fragment, it generates several, even billions of fragments. When just a part of the genome, needs to be investigated both for research and clinical application, it is known as target libraries. There are two methods commonly used for targeted approaches. One is what is called capture hybridization based sequencing. Other one is amplicon based sequencing, which are slightly more towards the life sciences. Only thing I just want to bring out is all these again require several software tools to complete the activity. Next, please. What I see, I don't know whether other viewers also see the same. Once the slide is displayed, the lower portion comes back again on top and covering the top portion. Okay, right. Next, let's get into what we call the second generation sequencing platforms. Second generation platforms belong to the group of cyclic array sequencing technologies. The technology that is used in second generation sequencing is called cyclic array sequencing. The basic workflow for second generation platforms include these things. One is the preparation of libraries. That's what we have seen in the previous slide. Then you need to amplify the libraries so that they turn out into billions of fragments. Then clonal expansion is a process by which you can clone this fragment. Then do the sequencing using the process of the NGS machine that you are using. And finally, the analysis. The analysis part could be more or less kind of common towards the end, but in between, in the primary and the secondary analysis, there is a difference between the analysis step that you need to follow with respect to ion torrent and Illumina. The two most widely used known sequencing companies of second generation sequencing are Illumina and ion torrent. Next, please. Illumina commercializes several integrated systems for the analysis of genetic variation and biological function that can be applied in multiple biological systems from agriculture to medicine. The process of Illumina sequencing is based on the sequencing by synthesis, SBS concept. It is called SBS technology. This is, capture on a solid surface of this, basically this technology involves capturing on a solid surface of individual molecules, followed by bridge PCR. PCR is the polymerase chain reaction that allows its amplification into small clusters of identical molecules. The cDNA is fragmented and adapters are added to both ends of the fragments. We have seen that adapters are added to identify the fragments. Next, each fragment is attached to the surface of the flow cell by means of oligos on the surface that have a nucleotide sequence complementary to the adapters, allowing the hybridization and the subsequent bridge amplification, forming a double strand bridge. This what happens in this step, and there are several processes here as you can see. Yeah, next please. This is about Illumina. This process, whatever we have seen in the earlier slide, this process is continuously repeated to generate several millions of dense clusters of double-stranded fragments, DNA, in each channel of the flow cell. The sequencing can thus occur by the addition to the template on the flow wall of a single labeled complementary DNTP, which serves as a reversible terminator. This again about the various processes. All that we need to understand is that it uses a mechanism called fluorescence, and there is it's based on the fluorescence. The sensors detect, and accordingly the sequence data is generated. Yeah, next please. This technology, Illumina technology, is considered to be highly accurate, and they claim that it can sequence up to 99.9%. accuracy. I think the slides are really disturbing too much. Is there a way that we can do something, Mr. Prasanna?
Sir, it's clear. It's clearly appearing, sir. For you, it may be the difference, but me, it's working properly.
Okay. Okay. Then I think we need to, because what's happening is it simply keeps jumping. Okay, fine. Anyway, we'll manage. Ion Torrent, the major competitor to Illumina, employs a distinct sequencing concept which is named as semiconductor sequencing. Basically, it makes use of CMOS technology to do the sequencing. The sequencing method in Ion Torrent is based on the change in the pH values caused by the release of hydrogen ions during the polymerization of the DNA. In Ion Torrent, instead of being attached to the surface of a flow cell, the fragmented molecules are bound to the surface of beads and amplified by emulsion PCR, generating beads with a clonally amplified target molecule. And one molecule per bead is generated in this process. Each bead is then dispersed into a microwell on the semiconductor sensor array chip, which is called, which is normally a CMOS chip. Yeah, next, please. Every time the polymerase incorporates the complementary nucleotide into the growing chain, the protein is released, causing pH value changes actually. The change in the pH value of the solution is detected by an ion sensor incorporated in the cell chip. These pH changes are converted into voltage signals, which are then used in base calling. Base calling is the process of identifying legitimate base pairs in the DNA. There are also limitations in this technology. The major source of sequencing errors are the occurrence of homopolymorphic template structures. During the sequencing, multiple incorporations of the same base on each strand will occur, generating the release of a higher concentration of hydrogen ions in a single flow. The increased shift in pH value generates a greater incorporation signal, indicating to the system that more than one nucleotide was incorporated. See, these are all biochemistry related things. And one interesting thing to note is that all these second generation machines, they make use of biochemistry, whereas the third generation machines, they make use of biophysics. That's why they are more efficient compared to second generation machines. Yeah, next, please. The third generation NGS technology brought the possibility of circumventing common transversal limitations of PCR-based methods. These limitations are like, nucleotide misincorporation, like what we have seen in the Ion Torrent in the previous slide. Then chimera formation, then allele dropouts, which is preferential amplification of one allele causing an artificial homozygosity call. The first commercial third generation sequencer was the platform Helicos genetic analysis system. This, in contrast to the second generation sequencers, the DNA was simply sheared, tailed, and there's a process called poly-A. And using this, the sequencing reaction occurs by the incorporation of the labeled nucleotides, which is captured by a camera. See, this is where, the biophysics comes in. With this technology, every strand is uniquely and independently sequenced. That's the real advantage of it. Next, please. In 2011, Pacific Biosystems introduced the concept of single molecule real-time sequencing with the PacBio RS2. It's SMRT, it is called. And a single molecule can be completely sequenced using this technology. This technology enables the sequence of long reads with average read lengths of up to 30 kilo bases. See, earlier second generation EPCs, they were able to sequence only to the tune of 500 bases or 1,500 bases in Illumina. Whereas here you can go up to 30,000 bases, read length. Individual DNA polymerase are attached to zero mode wavelength wells, which are this again a process. Maybe I think it is too much to get into our minds. I'll skip this. Basically, the next slide, please. Basically, it uses a camera to capture the thing. This technology enables de novo assembly and direct detection of haplotypes and high consensus accuracy. This allows epigenetic characterization, that is direct detection of DNA-based modification at one base level. Epigenetics is related to identification of the variances at one base pair level. The first sequences using SMRT technology face the drawbacks of having a limited high throughput, higher cost, and higher error rates. However, significant improvements have been made to overcome these limitations. More recently, PacBio launched a Sequel 2 system, which claims to reduce the project costs and timelines with high accuracy, and individual reads can be as high as 175,000 base pairs. Using a PacBio system, research demonstrated the successful application of long-read genome sequencing to identify a pathogenic variant in a patient with Mendelian disease, that is inherited diseases. This says that this technology has significant potential for especially the structural identification of the structural variation in the genes. Next, please. The second approach was followed by another company by name Oxford Nanopore Technologies, and they have released a machine called Mini Ion in 2015. It's basically a desktop technology, a desktop machine. This sequencer does not rely on SGS, but instead relies on the electrical charges in current as each nucleotide A, C, T, and G passes through the nanopore. These nucleotides pass through the nanopores, and they in turn they generate the electrical signals which are captured, and that's how the sequencing is done. And this where again we make use of biophysics rather than biochemistry. Nanopore sequencing uses electrophoresis to transport an unknown sample through a small opening. Then an ionic current passes through the nanopores, and the current is changed as the bases pass through the pore in different combinations. This information allows the identification of each molecule and to perform the sequencing. And in 2017, they released another model called GridION Mark I, which is a flexible benchtop sequencing machine. Yeah, and it can even generate next slide. It can generate. It can go up to 150 gigapairs of length. New advances were launched with Chromate Ion 48 system that offers 48 flow cells, and each flow cell can allow up to 3,000 nanopores. And this means that in about 72 hours, you can sequence about 7.6 tera bases. Longer reads are of utmost importance to unveil repetitive elements and complex sequences such as transposable elements, segmental duplications, and telomeric or centromeric regions that are difficult to address with short reads. These are all the problems that we are supposed to be solving with by making use of the NGS data analysis. This technology has already permitted the identification, annotation, and characterization of tens of thousands of structural variants from a human genome. Although the accuracy of nanopore sequencing is not yet comparable with that of short-read sequencing, for example, Illumina platform claims that their sequencing accuracy is of the order of 99.9%. It's not yet established in the case of third generation sequencers, this kind of accuracy. The updates are constant, and ongoing developments aim to develop the range of genomes and further improve accuracy of these things. And obviously, all these things are based on various software tools, and that's where the several challenges that we face come into play. The 10x Genomics was founded in 2012 and offers innovative solutions from single cell analysis to complex structural variant and copy number variant analysis. In 2016, 10x Genomics launched a Chromium platform. And in this technology, each gel, it makes use of a technology called gel beads in emulsion, and certainly, it's able to achieve quite a huge throughput when it comes to sequencing DNA. Next slide, please. For structural variant analysis, that is what is the kind of variation in the structure of the DNA is what we call structural variant analysis. The short-read libraries are computationally reconstructed to be able to perform megabase scale haplotype genome sequences using small amounts of input DNA. This technology allows linked read phasing of structural variant that distinguishes true structural variants from false predictions. This technology has the potential to be applied to de novo genome assembly. That is, you can assemble the genome from scratch using this technology. That's one big advantage. This can revamp difficult regions of the genome. It can detect rare alleles and can elucidate complex structural rearrangements within the genome. The Chromium system also offers single cell genome and transcriptional profiling, immune profiling, and analysis of chromatin accessibility at single cell resolution with low error rates and high throughput. This opens up exciting new applications, especially on the development of new techniques for epigenetics research. Epigenetics is primarily to look into the base pair level variations in the genome. Yeah, next please. Yeah. So what we have seen is about the second and third generation platforms, and today we have third generation platforms. However, the widely used platforms are still the second generation Illumina and Ion Torrent. And let's look briefly into what is involved in the NGS bioinformatics. Sequencing platforms are getting more efficient and productive, primarily because of the advancements in the technology. It is now possible to completely sequence the human genome within days at a relatively affordable price, which took years during 2001 to complete the human genome sequencing. And Promet Ion promises the delivery of human genomes for less than $1,000. It used to be quite huge when the thing started, and now in the last few years, the costs have fallen significantly. The amount of data generated demand the amount of data that we generate in any of this NGS sequencing. Like we talk of 3.2 billion base pairs, and the data about these many pairs results in huge data, and this is of the order of about 30 GB. The amount of data generated demand computational and bioinformatics skills to manage, analyze, and interpret the huge amount of NGS data. As long as now we know that how rich a human being is, how data rich rather, how data rich a human being is. Each cell has about 30 GB of data, even if you assume the base minimal thing, each cell, and we have about 3 trillion cells in our body. And this is only about the DNA. Apart from that, you have microbiology data and all other data. Considerable development in NGS bioinformatics is taking place, which could only take place greatly due to the increasing computational capacities. Basically, the improved computational capacities in terms of hardware, as well as the developments in various algorithms and applications to assist all these required steps. From the raw data processing to more detailed data analysis and interpretation of the variants in a clinical context, we need a combination of all these hardware and software. And because of this, we are in a better position today to do the NGS data analysis. NGS bioinformatics is subdivided into primary, secondary, and tertiary analysis. The overall goal of each analysis is basically the same regardless of the NGS platform that we use. For every platform has the same goal. Finally, at the end of the analysis, you should be able to come out with useful data that will be helpful in maybe treatment planning and prognosis and those kind of things. What it means is, at the end of the analysis, we should be able to find out what are the kind of variants, whether they are in small, maybe at one base pair level there is a variation, or at small sequence level, fragment level variation, or there are certain sequences, say, certain fragments which are repeated or deleted. These are all the things that we need to find out after doing the analysis. That this is the goal, irrespective of whether you use Illumina platform or Ion Torrent platform. The overall goal of each, okay, no problem. No problem, we can go to this. With like what we have seen in the previous slide, the NGS data analysis is divided into three parts, basically three stages. Whatever is shown in blue is the primary analysis, and the things that are done in secondary analysis is shaded with orange, and the green shaded region shows the tertiary analysis. Finally, what we get useful for our purpose is the output from the tertiary analysis. Of course, without performing primary and secondary analysis, we cannot do anything in tertiary analysis. Now, if you see the first step in the primary analysis is the signal analysis. Depending on the platform that we use, it has two parts. Briefly, we looked into each one of them. Illumina and Ion Torrent, they have two different processes for doing the signal analysis. These are all based on biochemistry. So one makes use of pH change converted into voltage. The other one makes use of image processing techniques. And once that is done, you do what is called a base calling. The base calling is the process of identifying legitimate base pairs in the DNA. And once this step is done, next step is now to do the base quality score in terms of a metric called pH red. This is a parameter which is used to assess the quality of the sequencing that has been done. Then at the end of this step, we generate a file called FASTQ. You get a FASTQ file. Then there will be certain reads that may be because of duplication, maybe the overlap and all those things. Therefore, there is a need for read filtering. After read filtering is done, and there is another process called demultiplexing. And after that, the adapters which were added before sequencing, they need to be trimmed. So there is a process what is called adapter trimming. Once all these things are done, you get a refined FASTQ file. So at the end of primary analysis, what you get is a FASTQ file, which is used in the secondary analysis for further analysis. And in the secondary analysis, what you do is you try to align the sequences that you got, which is in the form of FASTQ file, with the already known genome, like a human genome sequencing is what you're dealing with. You already have human genomes which have been standardized, which have been completed. The sequencing was completed in 2001, and further, there have been a lot of quality tests have been done, and they have been standardized. And in 2001, a reference genome was published, and in 2013, another reference genome is published. And then you align the sequences that you generated in the process of using the NGS machine and doing the primary analysis. You get the FASTQ file, you align the data that you get in the FASTQ file with respect to the reference genome, or if you are doing from scratch, if you are generating the genome from scratch, you do what is called a de novo assembly. Then this results in the generation of a file called BAM or SAM, that is binary I think we'll just see that it's basically binary alignment map. This file is generated at the end of sequence alignment with respect to already published genome or de novo assembly. After doing the alignment, then you do post alignment by removing. Again, there is a possibility that there could be certain duplicates of reads and read duplicates are removed. Then there is what is called a local read alignment, and then base quality scores which have been done in the beginning, that is as the third step, is again recalibrated based on the other process that has happened now. Then you look into the kind of variations that you find with respect to the reference genome and the genome that you have on hand, which was sequenced, done, has gone through the primary analysis and is going through the secondary analysis. You do what is called variant calling. Variant calling is the process of identification of the variation in the genome at either base pair level or small fragments level, or there could be multiple at multiple gene level also. It could be at different levels. So it is very, very crucial for us to figure out what kind of variants you have. And this results in generating a file called variant calling format file, VCF file. So what you had at the end of primary analysis is a FASTQ file, which is given as an input to the secondary analysis, which generates a VCF file. And the VCF file is used for finding the really end user useful data by using a processes called annotation, filtering, and validation and interpretation. And at the end of this process, you get a report. And the annotation involves functional annotation, where you look into the transcriptional gene regulations and alternative splicing. These are all the various things that one has to look into. And then again, you do a quality filter by making use of already published data. You compare and you filter certain things from a quality point of view, or over a period of time, if you are running a NGS data center, you can have an in-house database. You can use that as a reference, and then you can filter the the annotations that you have done, and this can be validated, and further interpretation needs to be done, which requires data visualization tools, etc. Next, please. So NGS bioinformatics, as we have seen, is divided into three. Yeah, I think we have already seen. Can you please go to the next one? Let's quickly look into primary analysis. The primary data analysis consists of detection and analysis of raw data, that is signal analysis, that's the first step. Targeting the generation of legible sequencing reads, that is called base calling, and scoring the base quality. These are three steps that we have seen. Then typical outputs from this primary analysis are FASTQ files, and the FASTQ file in Illumina, and it is a UBAM file in IronTorrent. Next, please. The principle for signal detection relies on fluorescence, and this is what we said, it makes use of biochemistry techniques. And the Illumina SPS technology delivers the highest percentage of error-free reads, and that's what the Illumina claims, that they can sequence up to 99.9% accuracy. Latest versions of Illumina's chemistry have been re-optimized to enable accurate base calling in difficult genome regions. Okay. Yeah, next, please. Yeah, I think this also we can. Okay. Now, if you take the Illumina platform, the older machines use a format called intermediate executable code for, in the case of older machines, INT they are called. See, base caller is one step in the primary analysis, And as I told, there are numerous algorithms available. For the base calling alone, you have large number of software packages available. And the base callers of Illumina can be divided into two. One is for the old platforms. Whatever is there at the bottom is for the old platforms. And whatever is there on top is for the new platforms. These are all the various softwares that are available, starting from AICTE, Rolexa, IBIS, etc. There are so many software that can be made use of. This is only for Illumina. Similarly, you have a big set for Ion Torrent. Next, please. See, in fact, there is a very popular algorithm used in these things, and all these algorithms make use of the so-called busted algorithm. And this is the most popular thing, and they claim that it reduces the error rates by 44 to 69% compared to other methods. Next one. And the primary analysis in the case of Ion Torrent is performed in the Ion Torrent software itself. Ion Torrent has its own suite of software, and the primary analysis is performed there. Starts with signal processing as usual, in which the signal of nucleotide incorporation is detected by the sensor at the bottom of the chip cell, converted to voltage and transferred from the sequencer to the server as a raw voltage data. This is named as a DAT file. So here again, you need a lot of software algorithms to come out with the DAT files. For each nucleotide flow, one acquisition file is generated that contains the raw signal measurement in each chip and each chip well for the given nucleotide flow. During the analysis pipeline, these raw signal measurements are converted into incorporation measures, and they are named as Wells files. See, all these things again, you have the raw data, you use certain algorithms to convert into Wells files. The base calling is the final step of primary analysis in Ion Torrent in this case, and is performed by a base caller module, which is also part of the Torrent suite of software. The objective in this case is to determine the most likely base sequence, the best match for the incorporation signal stored in the Wells file. That is the purpose. The mathematical models behind the base calling are complex and comprise 3 steps, which are listed here. Yeah, next please. Okay, this process is pictorially shown here. This for Ion Torrent, the workflow is shown like this. You have nucleotide incorporation, then the signals are measured, and by the sensors, it generates raw voltage file, and the raw voltage files are named as DAT files, and the server converts DAT files into Wells files, and then the Torrent base caller, which is part of the Torrent suite, identifies the bases, and then it creates a BAM file, that is binary alignment map file, which is used for further analysis. Yeah, next please. I think we skipped one slide in between. Yeah, okay. Anyway, we have seen, the BAM file is generated as part of the the Ion Torrent suite software output. Yeah, next please. See, this procedure is required to address some of the errors. Errors do occur, irrespective of the platform that you use, whether it is ion torrent or all Illumina, the errors do occur. These errors occur frequently, and thus, as an initial step, ion torrent performs phase correction and signal decay normalization algorithm. Again, the idea is to point out the kind of algorithms one has to work on to do in every step of this NGS data analysis. There are three parameters which are considered for this signal generation, which are called the carry forward, then incomplete extension, and group. These are all the things that are used. These are all two technical terms. Maybe I'll skip this portion. Yeah, next please. The CFIE and these three parameters that we have seen in the previous slide, the CFIE and DR measurements, as well as the normalized signals, are used by Solver. Solver is another software which is part of ion torrent suite. Follow the branch. This uses the branch and bound algorithm to do the further processing. The branch and bound algorithm consists of systematic listing of all partial sequences forming a root tree with a set of candidate solutions being placed as branches of this tree. The algorithm expands each branch by checking it against the optimal solution, that is the theoretical one, and goes on and on until finding the closer to the optimal solution. Before the first performance of the solver, there is a process called key normalization which is performed. The key normalization is based on the premise that the signal emitted during nucleotide incorporation is theoretically 1, and for the non-incorporation of the nucleotide, the signal emitted is 0. With this premise, this normalization is performed. Next, please. The key normalization scales the measurements by a constant factor, because instead of handling only 0 and 1, it performs a scaling. So to bring it to the signal to a known expected, one must produce by sequencing through the key as close to unit as possible. Once the solver has selected a base sequence, the predicted signal is used to create an adaptive normalization, which compares the theoretical signal with the real signal to subsequently generate an improved normalized signal with a reduced error rate. The error correction at every stage is important. Wherever there is an opportunity to correct anything, you go through the quality process. This enters in a cycle of subsequent iterations, applying a function repeatedly, with the output from one being the input of the next, with the solver for several rounds, allowing the normalized signal and the predicted signal to converge towards the best solution. This algorithm ends with a list of promising partial sequences. Next, please. Errors occur both in ion torrent and Illumina sequencing machines, and therefore it is very important to have a good quality control and good quality control process, and this is there throughout the NGS data analysis. The error, is expressed in terms of pH-red score, which is a logarithmic probability function. A pH-red score of 10, that is Q10, when they say, it means that 1 in 10 are likely to be incorrect. That is, what you have is an accuracy of 90%. When it is Q30, it means that 1 in 1,000. That is, you have the accuracy of about 99.9%. If it is Q40, it is going to be 1 in 10,000, which means it is has an accuracy of 99.99%. And let's say Q50, that means 1 in 100,000 inaccuracy is likely to happen. That means it's an accuracy of 99.999%. FASTQ files are important to the first quality control step, as they contain the raw sequencing reads, the file names, and the quality values with higher numbers indicative of higher quantities. The quality of the raw sequence is critical for the overall success of NGS analysis. Several bioinformatics tools were developed to evaluate the quality of raw data. The very well-known ones are NGS QC toolkit and FastQC, and these are all some of the well-known quality tools that are used at various stages of NGS data analysis. Next, please. The FastQC tool is FastQC is a very nice software, and this is the most popular among the quality tools that are available for NGS data analysis. As output, FastQC gives a report containing well-structured and graphically illustrated information about different aspects of the read quality. You get a very nice and user-friendly report from this. If sequence reads have enough quality, the sequences are ready to be aligned or mapped against the reference genome. That's very important. The sequence reads to be good quality one. The pH-rate score is also useful for filtering and trimming sequences, because this is a quality indicator. This score tells how good is the sequence reads that we have. Additional trimming is performed at the end of each read to remove adapter sequence that we have seen. It is part of because this was added before sequencing. The trimming step, although reduces the overall number and the length of the reads, that raises quality to acceptable levels, because they additionally added adapters have been removed, and therefore you're naturally bound to have less data on the same fragment. Several tools were developed to perform trimming with Illumina data. B-trim, Lee, Home, adapter removal, and Trimonotic. These are all some of the tools that are used with Illumina data. The choice of this tool is highly dependent on the data set, downstream analysis, and parameters that you use. It all depends on which tool you want to make use of. And in Ion Torrent, sequencing and data management are processed in Torrent Suite software, which has a Torrent browser as a web interface for data visualization and all. To further analyze the sequences, a demultiplexing process is required, which separates the sequencing reads into separate files. That's what is the process of demultiplexing according to the read. Next one. The demultiplexing is the adapter trimming step, whose function is to remove the remaining library adapter sequences from the end of the read. These library adapters were added before NGS sequencing was done to identify the beginning and end of the fragments. In most cases from 3' end, but can depend on the library preparation that you use. You would have seen the reads have the so-called 5' and 3'. So mostly these adapters are at the 3' end, and they are removed. This step is necessary since residual adapter sequences in the reads may interfere with the mapping and assembly. This is unnecessary. As such, it doesn't contribute for the DNA, and therefore there is no need for us to retain it, and therefore it is removed. Trimming is important in RNA sequence. SNP, that is single nucleotide polymorphism identification, and genome assembly procedures to increase the quality and reliability of the analysis and optimize the execution time and computational resources needed. Several tools are again used to perform the trimming with the Illumina data, and there is nothing like this the best tool that you need to make use of. There is no best tool. Instead, the choices depend on the downstream analysis. That is, what is that you plan to perform next, and what are the kind of parameters that you want to do a trade-off. These are the things that you need to consider. And this is also there, as part of Illumina, Ion Torrent. So, yeah, next one. Okay, what we have seen so far is about the primary analysis. The secondary analysis, 1 second. Madam, may I know how much more time do I have? Mr. Prasanna? Hello, am I audible?
Yes, sir, you are audible.
Okay. How much more time I have?
Hello.
Hello, madam. How much more time I have?
Actually, the next session starts at 11:30, sir.
Can I take another 5-10 minutes?
Okay, sir. Sure.
Okay, 10 minutes I'll take. 11:15 I'll wind up. Is that okay? I'm sorry about the initial hiccup.
It's okay. It's okay, sir. Okay,
Yeah.
sir. Fine.
Yeah, I'll quickly just only 1 or 2 more things I need to do. I'll just quickly finish. Yeah.
Okay. Okay.
Okay.
Fine. Thank
Yeah.
you.
Next slide, please. See, the purpose of secondary alignment, as we have seen in the previous diagram, no, it is basically the alignment of the reads with reference to a reference genome, or if you are doing a de novo assembly, it is to do a de novo alignment. Okay, next, please. Maybe I want to skip these things because we have seen a bit about these already. So I want to keep moving. Can you please, next, please. Next. In fact, all these places now we have several algorithms that now we'll be using. Next, please. Yeah. Okay. This is something I just want to spend a couple of seconds. These genome assemblers that we talk of, either they could be they're again, software pieces, software algorithms, and they could be either string-based assemblers or it could be graph-based assemblers. And string-based assemblers are greedy assemblers, and graph-based assemblers can be again divided into two, which are overlap layout consensus based and deep Bruegel graph. The deep Bruegel graph is type of a graph. So for each of them, there are several software packages available. Like for string-based assemblers, you have SAICTE, Shark CGS, VK, etc. And for OLC, you have CAP3, etc. There are so many such software packages which will be useful for secondary data analysis. Yeah, next, please. Next, please. Next. Next, please. Next. See, after the secondary data analysis, post-alignment processing is also important. That's what we have seen there. The quality process comes in again, and we mark the duplicates and remove them and things like that. And yeah, next, please. Next. Yeah, you can go to the pitfalls directly. NGS pitfalls. NGS pitfalls. Okay, yeah, please hold on. Yeah, there are several software which can be used. Functional filtering is one of the aspects of tertiary analysis, which we have seen as part of the annotation. And okay, we've seen sorting. There are softwares like PyLOP, then SIFT, Polyfen, CAD. These are all some of the softwares which are used for functional filtering. Next, please. Then you have Human Splice Finder, that is another software, NSSNP Analyzer, Topo SNP, Topographic Mapping NSP, Condel Consensus, Unaware. Unaware is a very, very popular software which does quite a good amount of bioinformatics work. It's very, very popular. Yeah, next one. Next, please. Web VEP is another variant effect predictor. That is also another very popular software. Unaware, Web. Next, please. Next. I think you have to go to the next one. Pitfalls. Yeah, I think you'll move one step back, please. One slide back, please. Yeah. See, it's quite some time before, after the human genome sequencing and the first NGS mission has been. No. It was okay. Pitfalls. Next slide. It's quite some time. It is 20 years now that after human genome sequencing, and also 17 years from the time the first. Next slide, please. From the time the first sequencer was introduced, but still we have a lot of challenges. And genomics field has grown greatly, and it allows the creation of the concepts like transcriptomics, genomics, metabolomics, etc. This information is very useful for us to understand the human health, no doubt about it. And NGS brought quite a lot of benefits and solutions, for especially the Mendelian diseases, and irrespective of the field, whether it is healthcare or agriculture or any other thing. Next slide, please. Next. However, it also brings in a lot of challenges. The first challenge is regarding the sequencing costs. Is it affordable to us today? Even if, let's say, somebody says that at $1,000, it's available today for sequencing on human one human. It's roughly about 75,000 rupees in the Indian context. And is it affordable by all the say 1.3 billion population that we have? It's a big thing that we need to see. Although the overall cost of NGS is coming down, and there are other costs involved which needs to be taken into account. And there are a lot of setup costs involved. Like setting up an NGS machine costs quite a huge amount. For example, an entry-level NGS machine costs about 2.5 crores, and the kind of the other things, consumables that you need to have, they also cost a lot. These are all some of the challenges that NGS has brought in, and typical exome sequencing of a human being generates about 30 GB of data, and the archival, storage, retrieval, processing, all those things are really issues. And added to all these things, there's a shortage of bioinformaticians who can do the analysis. Yeah. Next, please. Next, please. Yeah. I think you can. Yes. Okay, there are, though we have a lot of challenges like what were listed earlier, there are a lot of developments that are taking place, and especially the importance of bioinformatics is being felt in the country and elsewhere, and a lot of focus is on this area. And also the technologies like machine learning are coming into play a greater role in NGS data analysis, and these things are going to certainly help. Okay, I think I'll conclude with this. Thank you.
ನಾನು 55 years ಆದಾಗ ರಿಟೈರ್ ಆಗ್ಬಿಡ್ತೀನಿ. ಸೀಟ್ ನಾನು ಇದು ಪ್ರಕಾರ ಏನಾರ.
Thank you, sir.
Thank you. I don't know, in case anybody has any questions, they can you can forward them to me. Maybe I can specifically address them. I'm very sorry again for the initial hiccup. Anyway, I met the schedule, whatever has been set now anyway. Thank you.
Okay. Yes, dear participants, it's open for question and answer session. So if there are any doubts, means you can ask the resource person. Okay, fine. So it's my opportunity to conclude this particular session. So on bioinformatics and computational tools for next generating sequencing data analysis. So next generating sequence has transformed clinical genetic, making possible to analyze hundreds of genes, describing a DNA sequencing technology. So genetics, as you all know, it is important to medical practice as it provides definitive diagnosis for many clinically heterogeneous diseases. So this next generating sequencing or massively parallel sequencing, so which is used to designate a wide diversity of approaches. So here it was briefed about DNA sequencing timeline, and also sir briefed about the several brands on the next generating sequencing marketing. So a complete description about the second generation and third generation sequencing was given. So second generation sequencing is based on massive parallel sequencing or clonal amplification of molecules, and third generation sequencing is based on single molecule sequencing. So here the next generation sequencing library preparation steps was briefed about. So there are various methods of converting DNA into small fragments. So which there are different methods like acoustic sharing, sonication, hydrodynamic sharing, and enzymatic methods. So library, we can say it is a collection of DNA or RNA fragments which represent either entire genome or target region. So second generation sequencing platform, it consists of various steps like preparation of libraries, amplification of libraries, clonal expansion, sequencing, and analysis. And also it is discussed about the third generation. And NGS bioinformatics was mainly subdivided into primary, secondary, and tertiary analysis. And it is also briefed about the Illumina platform and Ion Torrent platform completely. The primary analysis workflow of both the platforms were discussed, and the genome assemblers such as string-based assemblers and graph-based assemblers were briefed about. Then at last it was discussed about the next generation sequencing pitfalls. Thank you for attending the session.
Thank you.
So we'll back at 11:30 for the next session. Thank you, sir, for the informative session. He's already logged out.